بسم الله الرحمن الرحيم. إن شاء الله الكل يكونون بخير وصحة وسلامة. This is lab 1 in urinary system. First we have to discuss the diseases of glomeruli in this lab. أول مرة أريد يعني الكل يعرف إنه إحنا when we examine or when we are dealing with a renal biopsy, we have to concentrate on 4 important items. The first is the glomeruli, number 2 tubules, the interstitium, and the blood vessels. In renal biopsy نحتاج ممكن اللايت مايكروسكوب، نحتاج الكترون مايكروسكوب، نحتاج immunofluorescence، نحتاج special stains. باللايت مايكروسكوب الروتين ستين مالتنا هي H&E hematoxylin and eosin stain. Sometimes we need special stains to examine the nature of the deposits, to highlight the sclerosis, the collagen, for example the amyloids or the sclerosis. We have the most important and the most commonly used special stains for renal biopsies هي ال PAS stain أو periodic acid shift stain. periodic acid shift ممكن نشوفني thickened basement membrane واللي قد يكون الكوز مالته يعني هايلينوسيس أو سكليروسيس. الفرق بين هايلينوسيس والسكليروسيس، الهايلينوسيس أو بالهايلين، هايلين هو عبارة عن acellular materials اللي هي mostly تكون glycoprotein، بينما السكليروسيس اكو fibrillar materials. هذه الفبريلار materials mostly ممكن تكون mesangial matrix، يعني increase mesangial matrix أو mesangial matrix يعني proliferation. الأذر سبيشال ستين المهمة هي سيلفر ستين. سيلفر ستين ممكن أشوف السكليروسيس أو عندي مثلا التركروم ستين. we can see the collagen deposition and the Congo red stain which is specific for amyloid. بالنسبة للإميونوفلورسنس، الإميونوفلورسنس هو يعني فد procedure to يعني مستفادين من خاصية الانتيجين antibody reaction. من يصير antigen antibody reaction, this reaction can emit a green light, mostly a green light when a special light is used, and يعني commonly هو UV light هو introduced. فمستفادين من antigen antibody when deposited in the glomeruli, it can emit a green light, so we use immunofluorescence to see this reaction. بالنسبة لل diseases of glomeruli, first we will start with the طبعا ممكن يكون nephrotic أو nephritic syndrome. وتعرفون النفروتيك syndrome والكوزز مالته اللي هي ممكن minimal change disease أو membranous glomerulonephritis, focal segmental glomerulosclerosis, membranoproliferative glomerulonephritis or other causes اللي هي ممكن يكون IgA nephropathy. أو النفرتيك syndrome اللي هي most commonly acute post-streptococcal glomerulonephritis و rapidly progressive أو crescentic glomerulonephritis. أول مرة خلي نشوف، أكيد أخذتوا بالهيستولوجي النورمال structure of glomeruli. The normal structure simply it consists of this Bowman capsule, which consists of the outer or the parietal layer and the inner layer or visceral layer of Bowman capsule, which is called the podocytes. In between we have the Bowman space, and here we have a meshwork of capillaries, and inside the glomeruli the cells which is called the intraglomerular cells or also called the mesangial cells. Here we have the efferent arterioles and the proximal tubules. Let's start with nephrotic syndrome. This is minimal change disease. It is exactly the same as the normal glomerulus. There is normal size, normal cellularity, no any change can be seen by light microscope. أكد إنه this is light microscope. in light يعني patient عنده nephrotic syndrome لكن الجلوميرولاي are normal, so this is minimal change disease. The size normal, the cellularity also normal. النفروتيك سيندروم الثاني هو focal segmental glomerulosclerosis. شنو focal شنو segmental؟ focal يعني part of the glomeruli, not all the glomeruli, some of the glomeruli are involved, not all the glomeruli. يعني أشوف glomerulus, this is normal and this is involved by the sclerosis. This is focal. Segmental means the glomerulus itself, part of it is involved by the sclerosis. So we see focal segmental, part of the some of the glomeruli, and the involved glomerulus partially involved by the disease. طبعا هنا there is a tubular atrophy. يعني اكو diseases affect glomerulus primarily and then affect the tubules, واكو diseases affect the tubules and then affect the glomeruli. So here, in addition to focal segmental glomerulosclerosis that involve the glomerulus, we have tubular atrophy. Here we have atrophied renal tubules. Now, focal segmental, as we mentioned, it is partially affect the glomeruli, and here we have deposition of sclerotic materials واللي ممكن تكون قلنا mesangial matrix proliferation. Here we use a special stain which is PAS stain, periodic acid shift stain, to show the sclerotic materials and partially effacement or obliteration of the Bowman space. Here there is no Bowman space in the part of the affected part. Membranous glomerulonephritis also can cause nephrotic syndrome. Here there is enlarged glomeruli. The cellularity are normal, only the size enlarged because of the diffuse thickening of the glomerular basement membrane. Here we have, this is H&E, hematoxylin and eosin. We can see here there is thickened basement membrane, and this will cause glomerular enlargement, but generally the cellularity are normal. Here we use a special stain which is a silver stain to see the thickened basement membrane here, which is stained dark. This is the thickened basement membrane, so it is membranous glomerulonephritis. In addition to that, we have spikes. Here we have the spikes that is shown by silver stain, so we have thickened glomerular basement membrane and the spikes. This is membranous glomerulonephritis, which is one of the causes of nephrotic syndrome. As we mentioned, we sometimes need immunofluorescence to see the green emission of the deposits. Here we have membranous glomerulonephritis, and we see here the deposition of IgG antibodies along the glomerular basement membrane. So this is immunofluorescence showing deposition of immunoglobulin G along the glomerular basement membrane. In IgA nephropathy, we can see, this is light microscope. Here we have a light microscope picture showing proliferation of the mesangial matrix. Here we have mesangial matrix proliferation, and this is important pathological change in IgA nephropathy. And here we have immunofluorescence that show deposition of IgA in the mesangial region. Here we have IgA deposition in the mesangial region by immunofluorescence, so it is IgA nephropathy. Now, nephritic syndrome. In nephritic, as we mentioned, most important causes are acute post-streptococcal glomerulonephritis and rapidly progressive or crescentic glomerulonephritis. Acute post-streptococcal glomerulonephritis here أول شيء عبارة عنه increase the size of the glomerulus and increase in the cellularity inside the glomerulus. Here the cellularity increased because we have many neutrophils, inflammatory infiltrate in the glomerulus. The stain used here is Mason trichrome stain. In rapidly progressive crescentic antiglomerular basement membrane glomerulonephritis, here we have a crescent formation. This is crescent formation, which is formed by increased in the cellularity of the especially the epithelial cells proliferation. So this is crescent, and the disease is rapidly progressive glomerulonephritis. Also, this is Mason trichrome stain. In hypertensive nephrosclerosis, here kidney is atrophied and the size is reduced. We have a congested red to brown color, and characteristically we have fine nodularity or granularity in the outer surface of the kidney in addition to atrophy. So this is hypertensive nephrosclerosis. Thank you.